So hello and welcome everybody to my channel Omni Bio Entrance, and today we will be seeing University of Hyderabad MSc Entrance Examination question paper for the subject Plant Biology and Biotechnology. I have discussed only the biology part. I have not attempted the chemistry questions. So coming to the first question. Which of the following systems of plant classification does not use botanical names above the level of order? So it is angiosperm phylogeny group. Answer is A. Stomata are completely absent in the plants which are submerged aquatics, because submerged plants do not have stomata and absorb nutrients and gases dissolved in water through the general surface. Then which among the following are sterilizing gases? So it is ethylene oxide and beta propiolactone vapor. So these are the sterilizing gases. So answer is option A. Then next one. Cyanogen bromide is a pseudo halogen compound with the formula CnBr is used to cut protein molecules at the C terminus of. So cyanogen bromide, it cuts protein molecule at the C terminus of methionine residues. Answer is option D. Then considering a cross, they have given the genotype with all the genes independently assorting the proportion of progeny that will phenotypically resemble the first parent. So what portion of the progeny will phenotypically represent the first parent? I have not shown all the options because I want to put this photo over here. Okay, so I have not shown all the options. Anyway, the D option is not the correct answer. Okay, so we will see. So you have capital A small a and it is crossed with small a small a. So you will get a probability of capital A small a, as the genotype in the progeny is capital A small a, it is half. Then if it is capital B into capital B small b, then probability of getting capital B is half. Then if it is like the cross between capital C into small c, then the probability of getting capital C small c is half. Similarly, in the case of D, it is half, and in the case of E also it is half. Now, if you multiply everything, that is half into half into half into half into half, so 1 by 32. So that is the proportion of progeny that will phenotypically resemble the first parent. So answer is option A, that is 1 by 32. A similar question was asked in the last year examination also. Then when bacteria are grown on medium containing 35S as the source of sulfur, 35S gets accumulated in. So it gets accumulated in protein, because only proteins have sulfur in them, all the others do not have sulfur. So sulfur containing proteins are present, whereas DNA, RNA and mRNA does not have sulfur. Then for the peptide bond, which is not true, there is perfectly free rotation about the peptide bond. Because of the partial double bond between alpha carbon atom and the amino nitrogen atom, no rotation is possible around the bond. So option A itself is the right answer, and it is not true with regard to peptide bond. Then next one. Eye color of Drosophila melanogaster is sex linked, with red eye color being dominant to white eye color. In a cross between a red-eyed male and a heterozygous female, all of the following progeny are expected except. So a cross is made between red-eyed male. So X, R and Y. So that is red eye, and X small r is white eye. So red-eyed male, that is heterozygous. So X, R and Y, and a heterozygous female. So X, R, X small r. Okay, these are the crosses that you will get from these gametes. So what they have asked, so in a cross between red eye male and heterozygous female, all of the following progeny are expected except. So this is red eyed female. So that's true, you will get it. Then red eyed fertile males. Red eyed fertile males, XRY, so that is also true. Then white eyed fertile males. white-eyed, yes, X small r y, that is also true. White-eyed fertile females. You will not get white-eyed females, it will be red-eyed. So answer is option D. Then next one. Maintaining the normal glucose level is very important for normal function of cell. Insulin is very important hormone and has a vital role in maintaining blood sugar level. The process of maintaining blood glucose level through degradation of glycogen is known as. So, glycogenolysis. Answer is option A. So, glycogenolysis, that is a degradation of glycogen for maintaining blood glucose levels. Then what is gluconeogenesis? That is synthesis of new glucose from non-carbohydrate precursors. It provides glucose when dietary intake is insufficient or absent. Then what is glycolysis? The breakdown of glucose by enzymes releasing energy and pyruvic acid. And glycogenesis, the process of glycogen synthesis in which glucose molecules are added to chain of glycogen for storage. Then next one. The transfer of undesirable traits through introgressive hybridization from parent to offspring is resulting from. So it is resulting from linkage drag. So linkage drag, it refers to reduction in fitness in a cultivar due to deleterious gene introduced along with the beneficial gene during backcrossing. So the transfer of undesirable trait through introgressive hybridization from parents to offspring is resulting from linkage drag. Answer is option A. Then next one. The progeny of a single self-fertilized homozygous individual is known as. It is called as pure line. Answer is option B. So plants are considered inbred when the offspring, that is progeny, from a self-fertilized plant look identical to the parent as well as identical to one another. Then clone, it is a group of individuals of like genotypic constitution. And pure line, it is a group of individuals traceable through solely self-fertile in a single homozygous ancestor. And hybrid, you all know what is a hybrid. Then next one. The physical co-localization of genetic loci on the same chromosome within an individual or species is known as. It is known as synteny. Answer is option C. Then iron and magnesium are needed for. So iron and magnesium are needed for synthesis of chlorophyll. Option D. Identify the mismatch between the type of enzyme and its example. So oxidoreductase, lactate dehydrogenase, hydrolase, glucose 6-phosphatase, ligase, glutamate synthetase, and transferase fumarate hydratase. So fumarate hydratase, it belongs to hydrolases. It catalyzes the reversible hydration or dehydration of fumarate to malate. So answer is option D, that is the mismatch. Then the presence of catalyst in a reaction mixture can affect the activation energy. It lowers the activation energy. Then Voges-Prosker test is a procedure that detects the presence of acetoin. Answer is option C. Chromosomes primarily consist of DNA and proteins. These proteins are of two major classes called histones and non-histone chromosomal proteins. Histone proteins are rich in which amino acid? So the question is, histone proteins are rich in which amino acids? So histone proteins are rich in lysine and arginine amino acid. Answer is option B. Which is the sunshine hormone? So it is vitamin D that is called as the sunshine hormone. Answer is option A. Then next one. The allelic variants or form of an enzyme which differ structurally but not functionally are known as. They are known as allozymes. Answer is option A. Isozymes are enzymes that differ in amino acid sequence but catalyze the same chemical reaction. And holoenzyme, it is a complete functional enzyme which is catalytically active. And holoenzyme, it consists of apozyme together with its cofactors. Now, which among the following is the most appropriate equation for nitrogen fixation? It is a repeated question. So, answer is N2 plus 8H plus 8 electrons plus 16 ATP giving 2 NH3 plus H2 plus 16 ADP plus 16 Pi. So, answer is option D. Then next one. A ribonucleic acid that catalyzes a chemical reaction in a cell in a similar way to that of an enzyme is known as, it is called as ribozyme. Answer is option C. And what is abzyme? Abzyme is monoclonal antibody with catalytic activity. And synzymes, they are substances with catalytic capabilities. Then, which of the following statements is not true for inulin? So, inulin, it is a carbohydrate composed of 1 glucose residue and 2 to 60 fructose residue linked via beta 2,1 glycosidic bond. So, here it is a storage carbohydrate found in bulb of many plants. That is correct. It contains fructofuranose units joined together. That is also correct. It is a protein highly glycosylated. That is not correct. It is a structural carbohydrate containing lignin. That is also not correct. So, answer is option C, that is R and S. The first time opening of a flower or a spikelet in a spike is known as, it is called as anthesis. Answer is option B. And prosthesis, that is an artificial body part such as a limb, a heart, or even a breast implant. Then next one, Stickland reaction. So this is also a repeated question. It is between two amino acids in which one amino acid is oxidized and second amino acids act as an electron acceptor. So answer is option A. Then next one. The proportion of total variance contributed by the genotype of an organism is called. So it is called as heritability. Answer is option D. Then genetic advance or response to selection is a measure of how much gain you may get from phenotypic selection of a trait. Then selection differential is the difference of the base population mean and the mean of the selected parent. The selection response is how much gain you make when mating the selected parent. And panmixis is random mating within a breeding population. Next one. The largest family of angiosperm in the world is, it is Asteraceae. Answer is option B. Then genesiogamy is a type of pollination where transfer of pollen grains from the anther to the stigma take place between different flowers in the same plant or genetically similar plant. Answer is option A. So coming to the next question. In proto-cooperation, two organisms get mutually benefited and the relationship is. So the relationship is not obligatory. Answer is option B. Then identify the mismatch from the following. So the mismatch is oligophile, that is they have given as pressure, but actually oligophiles are able to grow at extremely low concentration of nutrients. Then after agarose gel electrophoresis of plasmid, which form of plasmid follow the sequence in decreasing molecular weight? So for your agarose gel, you have got the gel like this. They are the wells. Then here you have got the wells. Okay. And then you will load the marker in one of the well and the plasmids in the other. So the marker here, it will be the maximum size of the marker, that is 10 kb if you are using a 10 kb marker, and the lowest will be around here, that is at the base of the gel, that is 1 kb. So now if you load a plasmid, so where will this linear plasmid, open circular and supercoiled plasmid will be? That is what they have asked. Since the supercoiled plasmid, it is the supercoiled form of DNA structure, it will migrate and run to the base of the plasmid, that is over here you will get the base of the agarose gel, that is it has got less resistance to run through the gel, so it will run and you will get the band over here. Then comes the linear plasmid and the open circular. And out of these two, the linear plasmid will be somewhere around here, and the open circular will be at the topmost region. Open circular is one in which there is only one double stranded portion, that is only a single stranded cut is made. Okay, not both the strands are cut. Now in the case of linear, both the double strands are cut with the restriction endonuclease. So they will be linearized plasmid. That will be over here, and open circular will be over here. So the maximum molecular, so decreasing molecular weight they have asked. So the lowest molecular weight is for the supercoiled one and highest is for the open circular one. So the answer is open circular, then comes linear plasmid, and then comes the supercoiled plasmid. Okay, so the answer is option D. That is open circular, linear plasmid, and then comes the supercoiled plasmid. Answer is option D. Then cone has a diploid number of 20. How many chromosomes would be expected in a meiotic product? A polar nucleus, a mature embryo sac, and an endosperm cell. So polar nucleus is haploid, meiotic product is haploid, and endosperm is triploid. So it will be 10, 10, 30, 30. Then next one. A species of plant is discovered in which individual plants produce either white or purple flowers. True breeding plants that have short stems with white flowers are crossed with true breeding plants that have tall stems and purple flowers. The resulting offspring all have tall with purple flowers. When one of these tall purple flowered offspring is crossed with a short white flowered plant, plants with the following characteristics were obtained in equal proportion. White flowers on tall stems, purple flowers on tall stems, white flowers on short stems, and purple flowers on short stems. Given the information above, which one of the following is most likely to be true? So of these, the gene for flower color and stem length are not linked. They are independently assorting. This is the same as the dihybrid cross which Mendel carried out. So this is the question that they have asked, that is, you are crossing a short stem white flowered plant with a tall stem purple flowers. Then you get all tall with purple flowers, similar to Mendelian dihybrid cross. Now you cross the tall purple with short white, that is, you cross the F1 progeny with the homozygous recessive, and it is called as the test cross. And you will get tall white, tall purple, short white, and short purple plants. They are in the ratio of 1:1:1:1. So they show independent assortment. So the genes for flower color and stem length are not linked. If linkage was there, then you would not have got this ratio. Then next one. Which of the following statement is not consistent with the principle of totipotency? So cell specialization is based on gene content. So answer is option D. So cell specialization or modification or differentiation, it is a process that occurs after cell division where the newly formed cells are structurally modified so that they can perform their function efficiently and effectively. So which is not consistent with the principle of totipotency is cell specialization is based on gene content. So that is the wrong answer here. Is the incorrect statement here and is the right answer. Then DNA methyl transferase adds the methyl group to the fifth position of cytosine. Answer is option B. Then polycistronic mRNA refers to. So polycistronic mRNA is mRNA that is translated by many ribosomes simultaneously. Or it can be even mRNA with multiple open reading frames. So answer is both C and D. Both C and D are correct. Then next one. The calcium is very essential element for organisms' growth and development. Change in Ca2 positive concentration can initiate various responses in plants. Which one of the following responses is not known to be initiated by change in Ca2 plus concentration? So closure of stomata. Now closure of stomata, it is potassium, not Ca2 plus ions. So guard cells accumulate K plus, which results in opening of stomata, and release of K plus, which result in closing of the stomata. So answer is option A, that is closure of stomata. So it's not driven by Ca2 positive ions. Then next one. Due to high level pollution, the gases sulfur dioxide, SO2 and carbon dioxide, CO2 concentration increased drastically. It can cause the acid precipitation. The acid precipitation has lowered the pH of a particular lake to 4. What is the hydrogen ion concentration of the lake? So they have given the pH of the lake is 4, and pH is equal to minus log concentration of H plus or minus log 4. So that will be the hydrogen ion concentration, then will be 10 raised to minus 4. So answer is option A, 10 raised to minus 4 molar. So that is the hydrogen ion concentration of the lake. Then next one. If the coding region of a gene is estimated to consist of nucleotides of 300 base pairs, how many amino acids would be the corresponding polypeptide chain? So coding region is 300 base pairs. So 3 base pairs will code for 1 codon, and 300 base pair will code for 100 codons. So answer is option D, 100. Then symbiotic biological nitrogen fixation takes place with association between a plant and a nitrogen fixing prokaryote as shown in the following table. So list of plants and then nitrogen fixing prokaryote. Glycine max. Glycine max is Bradyrhizobium A2. Then Datsika. Datsika is Frankia. Gunnera, it is nostoc, and Azolla, it is Anabina. So answer is option B, that is A2, B5, C4, C6, D3. Next one. Which of the following plant is best suitable for in situ phytoremediation of heavy metals in an inland pond or lake? So water hyacinth. Answer is option A. After pollination, which of the following events is vital for fertilization to occur in flowering plants? So a pollen tube grows from the stigma to the ovule. Answer is option D. When a diploid embryo sac is formed from a megaspore mother cell without a regular meiotic division, the process is called, it is called diplospory. Answer is option D. Parthenogamy, it is the union of two incompatible gametes, that is fusion of two female gametes, is an example. Then polyspermy, it is fertilization of an ovum by more than one sperm, and polygamy, it is the condition of bearing some same flowers with stamens, some flowers with stamens only and some with pistils only, and born with both on the same or different plants. So that is polygamy. So here the answer is diplospory. And next one. The phenomenon leading to the development of embryo from synergids or antipodal cells without fertilization is known as apogamy. Answer is option B. And what is apospory? The gametophyte develops from the 2n sporophyte directly, and autogamy, it is self-pollination. Then match the following research institutes with correct places. So Bose Institute is Kolkata, Indian Institute of Science IISc, it is in Bangalore, Indian Agricultural Research Institute C2, that is New Delhi, and National Institute of Nutrition NIN, it is in Hyderabad. So the answer is option B, A4, B3, C2 and D5. Next one, Okazaki fragments are formed on. So Okazaki fragments, they are formed on the lagging strand of DNA during DNA replication. Find the correct match of Nobel Prize and section. So Jeffrey C. Hall, Michael Robash, Michael W. Young. They were awarded Nobel Prize for 2017 medicine, A4. Then Dennis Mukwege, Nadia Murad. They were given Nobel Prize in 2018 for peace. Francis H. Arnold, George P. Smith and Sir Gregory P. Winter. They were awarded Nobel Prize for 2018 chemistry. Then Arthur Ashkin, Gerard Mourou and Donna Strickland. They were given in 2018 physics. So the answer is A4, B6, C2 and so coming to the first question, match the following. Dendrobium. Dendrobium, it belongs to family Orchidaceae, A3. Then Tradescantia. Tradescantia is B4, that is Comilinaceae. And Fragaria is C1, that is Rosaceae and Syzygium is Myrtaceae. So answer is option D, that is A3, B4, C1 and D6. So answer is option D. Then cultivated bananas, they are sterile because they are triploid and therefore seeds are not set. Answer is option C. Then match the type of ovules with the corresponding plant. Orthotropous. Orthotropous is seen in the polygonum type, A3. Anatropous. Anatropous is for Ricinus. Amphitrophous is for Lemma and Cercinotrophous is for Opuntia. Answer is option C, that is A3, B4, C1 and D2. Dieback of shoots disease is caused by the deficiency of copper. Answer is option C. Next one. Which one of the following is an anapleuritic reaction? So the anapleuritic reaction is anabolic reaction that help to generate the intermediate compounds of the biochemical pathways. So conversion of pyruvate to oxaloacetate is an anapleuritic reaction. Answer is option D. Then next one. Urease. Urease, it requires which ion? That is nickel, Ni2 plus, A2. Then DNA polymerase, it requires Mg2 plus and nitrogenase, it requires molybdenum, MO plus 3 and carbonic anhydrase, it requires Zn plus. So the answer is option B, that is A2, B6, C4 and D5. Then find the appropriate match. Beta oxidation. So beta oxidation, it takes place in mitochondria. So A6. Then 50S ribosome. 50S ribosome is for smooth endoplasmic reticulum. Light reactions, they take place in chloroplast and steroid biosynthesis, it takes place in peroxisome. So answer is option A, that is A4, B3, C, A6, okay, A6, B3, C4 and D2. Next one. In angiosperm, the ABC model pertains to flower development. Answer is option C. In Drosophila, which of the following types of chromatin modification is associated with dosage compensation? So histone H4 acetylation. Answer is option A. To amplify the DNA sequence by PCR, which of the following primer sets would you use? So the answer is option B, that is TAG, AA, GTA, TGC and TAG, AA, GC, TA, TGC and GCG, GCC, GC, AA, GC, TT3. So these are the primer set that you would use to amplify this particular DNA sequence that is complementary to this. So TAG is complementary to ATC, TTG, AAG, TA, then comes TAT and TGC. Then you have got AAGC, TTG. So the complement of or the reverse complement, that is from 5' to 3' direction. So you have to read in this particular direction. That is so you have to write the complement in this direction. Here it is 5', here it will be 3'. Okay, so the answer is GC, CG, CAA, GC, TT. So that is the primers that you require to amplify this particular fragment. Then next one, presence of vessel-less wood with the tricolpate pollen grains in tetracentron is an example of. It is an example of heterobathmy. Homoplasy is the character shared by a set of species but not present in their common ancestor. And parallelism, an evolutionary process by which two or more separate species in the same environment develop similar adaptation or characteristic for survival supplement. Then next one, a natural silk is a polymer derived from, it is derived from amino acids. Answer is option A. The element that acts as a cellular cation and is involved in osmotic regulation is, it is potassium. Answer is B. The radioactive isotope of hydrogen is, it is tritium. Answer is option C. Then the statements is or are true for glycocalyx. So highly fibrous network of carbohydrates that cover the membrane of cells, bacteria. Then calcium deposit on the surface of the cells, lipid soluble compounds present on the cell surface, and it is not present in plant cells. So the statement that is true for glycocalyx is only P. It is a highly fibrous network of carbohydrate that covers the membrane of cells of many bacteria. So answer is only P. Then which one among the following inhibits the cell wall synthesis? So methicillin, it is a semi-synthetic derivative of penicillin. So that is involved in inhibition of cell wall synthesis. One among the following is not a food preservative. So cinnamic acid, given here, it is not a food preservative. Then match the following. Loktak Lake. Loktak Lake is Ramsar site. So A2. Then Ghana. Ghana is bird sanctuary. Ghana is for a national park. And Nokrek, it is biosphere reserve. So answer is option A. A2, B6, C4, D3. All the plant groups possesses phloem parenchyma except. So monocots don't have phloem parenchyma. Answer is option B. Then fermentation, a word with many meanings for a microbiologist and for a biochemist. It is use of organic substrate as the electron donor and acceptor. Answer is option B. Then a seed protein was isolated and purified in the purification method used for purification material Cephedex G150. Which of the technique used in above purification? So it is gel filtration chromatography. In that you will use Cephedex G50. Then next one. Which of the plant cell live cell type is devoid of nucleus at maturity? So it is sieve tube elements that is devoid of nucleus at maturity. Answer is option D. So coming to the next question. Asafoetida. Asafoetida is Ferula species A6. Then comes Santonin. Santonin is Artemisia maritime. Resveratrol is Vitis vinifera and Caryophyllene is Syzygium aromaticum. So answer is option D, that is A4, A6, B4, C2 and D1. So next question, which is the Michaelis-Menten graph for competitive inhibition? So it is graph R, that is an example that is showing competitive inhibition. So answer is option A, because in the case of competitive inhibition, the Vmax remains the same and Km increases. So competitive inhibition is when the inhibitor binds to the substrate binding site and will not allow the substrate to bind to the active site or substrate binding site. So in that case, the Vmax remains the same but Km increases. So answer is graph R. Then what is C value? C value, this is the amount of DNA contained within haploid nucleus of an organism. So answer is option D. Then agarose gel electrophoresis is used for DNA RNA detection. What is the nature of this agarose and from where it is obtained? So it is a polysaccharide in nature and it is obtained from red alga. So answer is option C. Then which of the following technique is not related to gene expression analysis? So AFLP is a PCR based technique that uses selective amplification of a subset of digested DNA fragments to generate and compare unique fingerprints for genome of interest. So it is not related to gene expression analysis. Northern blot is related, real-time PCR is involved and microarray is also involved in gene expression analysis. And next one, in which of the following techniques restriction endonuclease is not used? So it is random amplified polymorphic DNA or RAPD. Restriction endonucleases are not used. Then what is alluron layer and what is its nature? So it is the outermost layer of endosperm and is made up of protein. Answer is option A. The male sex organ of bryophyte is called as antheridium and archegonium is the female sex organ. So answer is option B. Then what is perination? It is the ability to survive from one germinating season to another, especially under unfavorable conditions. So answer is option A. Then in which of the following experiment a probe is required? So in the case of southern hybridization, you require a probe. Which of the amino acids oxidative reductive state determines a protein migration on SDS page? So it is cysteine. This is a repeated question. I have already discussed it in the previous question papers. So you can go through it. Then next one. Match the commercial microbial source in group 1 with the products in group 2. So Corynebacterium lilium, it is glutamic acid A6. Then Klebsiella oxytoca, it is 2,3-butane-diol. And Aspergillus niger, it is citric acid. And Alkaligens eutrophus, it is poly-beta-hydroxybutyric acid. So answer is option B, that is A6, B3, C4 and D2. Then which of the following are considered to be the best source material for in vitro mutant isolation in plants? So cell suspension cultures. Answer is option C. So coming to the next question. All of the following are true about plant growth regulators except. So specific organs are involved in the synthesis of plant growth regulators. Answer is option C, that is not correct with regards to plant growth regulators. Then sugar beet is diploid with 2n is equal to 18 chromosomes. How many chromosomes will be found in monosomic mutant of this species? So 2n minus 1, that is 18 minus 1, that is 17. Then a certain species of plant produces flowers with petals that are normally blue. Plant with a nuclear mutation small w produces white petals in homozygous condition. So plant is normally blue means that it is the dominant trait is blue. So capital W. And plant with the nuclear mutation. So if a mutation takes place, it is producing white petals. So small w will give rise to white petals and in homozygous condition. So in a plant of genotype small w, that is homozygous condition, that is white. Okay, white flowered plant, 1 W allele reverts to wild type during early stages of plant development. So out of this, that is out of small w, 1 W is reverting to wild type, that is capital W. So you will get the plant with capital W, small w. The detectable outcome that this reversion produces in the resulting petal is, it is blue petals because capital W is dominant over the small w. So blue petals is the. Then match the entries in group 1 with the elution condition in group 2. Ion exchange chromatography. Ion exchange chromatography is increasing gradient of salt, A5. Hydrophobic column chromatography, decreasing gradient of polarity. Gel filtration chromatography. Gel filtration chromatography is isocratic solvent and chromatofocusing is ampholytes. So answer is option C. A5, B4, C1 and D3. Then next one. Match the following. Acid value. Acid value is A3, that is free fatty acid. Biuret test is protein, B6. Kilani synthesis is monosaccharides. And Benedict's test is for reducing sugars. So answer is option A, that is A3, B6, C5, D2. Then what are the possible peptides obtained by digestion with trypsin? So trypsin cut the peptide chain mainly at the carboxyl side of amino acid lysine and arginine. Okay. So this is K is lysine and R is arginine. So here this is the polypeptide sequence that is given, and we have to see at how many what are the digestion products of trypsin. So here ASK, so it will cut at the carboxyl side of K because K is lysine. Then you again go forward, you will find another K, that is lysine. So again it will cut. Then here there are no K or R present, so it will not cut. So you will get 3 fragments. So this is the amino end and this is the carboxy terminal end. So NH3 ASK CO minus, NH3 YSATTPY ESL PK TYW CO minus, and NH3 plus LYPP ASTK CO minus. So these are the fragments that you will get upon digestion with trypsin. So answer is option B. Then next one, pseudo dominance. Pseudo dominance, the expression of recessive mutation in a heterozygous individual in the absence of wild type allele. Answer is option A. The transfer of gene from one organism to another or transfer of genetic variation from one population to another, it is known as gene flow. Answer is option A. Gene conversion, the process by which one DNA sequence replaces a homologous sequence that the sequence become identical after the conversion event. Then gene substitution, it is a type of mutation where one base pair is replaced by a different base pair. And gene redundancy, it is a term usually used to describe situations where a given biochemical function is redundantly encoded by 2 or more genes. Then expansins are involved in So they are involved in cross-linking network in cell walls. Answer is option D. Then what is the correct order of inflorescence? So this is the case of compound umbel. In compound umbel, all the umbel inflorescence arise from a common point and appear to be at about the same level. So in the case of umbel inflorescence, all the umbel inflorescences arise from a common point. This is the common point and it ends up in an umbel inflorescence. See, like this. And these are all individual umbel inflorescences, and this is the compound umbel. Then third inflorescence has the main axis that grows indefinitely and the sub-axis have determinate growth. So this is the axis that grows indefinitely. That is why an arrow is given over there. This axis grows indefinitely, whereas these sub-axis have determinate growth. Then the next one is a compound raceme, when the main axis of the inflorescence is branched and the lateral branches bear flowers in a simple racemose manner. That is the case of a compound raceme. So this is a case of a compound raceme. So the main axis is branched and the lateral branches it bears flowers in a simple racemose manner. So that is in the case of a compound raceme. Then you have got a compound corymb. Here the main axis, that is peduncle, branches in a corymbose manner and each branch bears flowers arranged in corymb. So that is called as compound corymb. So the answer is C, that is compound umbel, third compound raceme and compound corymb. So answer is option C. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.